Hello friends, so to start with a very interesting topic, that is cDNA synthesis, and everyone knows that step because we have used a diagnostic test for during, or we have learned the PCR or real-time PCR during COVID-19. So in molecular laboratory, cDNA synthesis is very common process which we used to use for gene expression studies or for various and many other experiments. So for the cDNA synthesis, there are some common steps. cDNA means complementary DNA synthesis. So there are very common steps. There are different or common steps. The first one is your RNA sample or RNA extraction. RNA extraction, that as we have discussed in previous lecture, that RNA can be extracted from different type of samples like tissue sample or cell line or your even nasopharyngeal swab samples or your any body fluid, blood, and many more samples we can use for the RNA extraction. The RNA extraction is very important and crucial step. Why it is crucial? Because if the quality of your RNA is good or it is maintained, then the quality of cDNA and the further is downstream application. If you want to go with the gene expression studies, or if you want to prepare cDNA libraries, or if you want to go with the even next generation sequencing, that means RNA extraction is very important. So we should focus the quality on the quality of RNA extraction. RNA extraction, there are different procedures for extraction of the RNA from the different samples that we have discussed. There are there are manual methods for the RNA extraction, and there are kit-based RNA extraction methods. So what precautions you have to take? You have to process your sample at 4 degree, or you have to use ice, crushed ice, for the extraction of RNA. Second is, if you are working with the kit, even then you have to take some precautions, like you have to your environment should be RNase free, and your gel apparatus, where you are checking the quality of RNA, or that should be DEPC treated. That water should, that those apparatuses are treated with the DEPC water, and just to get remove of all the RNases from the apparatuses, and all many precautions you have to consider while you are working with the RNA extraction. So if I consider that your RNA extraction or RNA quality of RNA is maintained in your laboratory, then it's easy to work with the cDNA synthesis. So the first step is RNA extraction, and then the second step is you have to remove your DNA. You have to remove your DNA contaminants. DNA contaminants. As in previous lecture, I have explained very nicely how you can remove your DNA from your RNA sample, because that is the major contaminant in your RNA sample. And even in nanomoles or even in femtomoles, the RNA is contaminated with your DNA. So whenever you are you are comparing, or you are interested in relative expression studies, or even in viral load studies, or even gene expression studies, or even cDNA synthesis. So in all those cases, or if you are going with the diagnostic, then you have to remove your DNA from the your RNA samples. And one more thing is, DNA contaminant can be removed by, or it is removed by DNase treatment. And there are other protocols also to remove your DNA molecule, but the main is your you can treat your RNA sample with the DNase one, just to remove your DNA from the. And even if you check the quality of RNA should be maintained after treating your RNA sample with the DNase one. So DNA should be removed from your sample. One very interesting thing I want to explain here is that, suppose if you are working with your prokaryotic RNA, or even if you are working with the eukaryotic or even human samples. In human, you have exon and introns. So Suppose this is your exon, exon 1, exon 1, exon 2. You know that if you design primer within exon for your PCR, then you cannot differentiate your DNA and your RNA. What I have told, if you have designed your primer within exon, then you cannot differentiate your DNA or RNA sample. But if you design your one primer in exon 1 and second primer in exon 2, then there are chances you can differentiate DNA versus your RNA molecules. But whenever you are working with the RNA samples where exons are not, where only you have exonic part, you don't have any intronic part, in those cases like viruses, in that case you don't have any choice to design your primer, so you have to design in one exonic part. So in those cases, if you want to differentiate your DNA and your RNA, and that in those cases you should treat your DNA or RNA sample with the DNA swan. This is your second step. The third very interesting step is you have to select your reverse transcriptase RT enzyme. Why I am telling this? Reverse transcriptase. In initial days, the cDNA synthesis was carried out by avian myeloblastosis, the virus, reverse transcriptase isolated from that virus. And what are we should know the what are the differences and what is our priority, which reverse transcriptase we want to use. So, to hold on this third step, we should know different type of reverse transcriptases. So, we'll go with the reverse types of reverse transcriptases. So, it's interesting, and we should know that. So, there are three type of. One is AMV, avian myeloblastosis virus reverse transcriptase. Other one is MMLV, Mulliny-Murine leukemia virus reverse transcriptase RT, and this is RT from this. And the third one is, this is second one, this is third one is modified or recombinant. You can say recombinant MMLV reverse transcriptase. So these are the three different type of reverse transcriptases. We should know the difference in all the three. So the first difference is this AMV. This is the first reverse transcriptase which was you can say discovered or utilized in laboratories for the conversion of cDNA. So the stability, this enzyme is highly stable. The stability, this enzyme is stable. This is less stable, and this is, you can say, more stable. The second difference is it can convert less than 5 kb. It can convert less than 7 kb, means little longer RNA molecule can be converted to cDNA with the help of MMLV, and with this you can go with the 11 or 13 kb. Right? This enzyme works at 42 degree. This enzyme works at 37 degree, and this enzyme can work at 55 degrees Celsius. So if your application or if your downstream application or cDNA synthesis you want to carry out at 42 or 37, 55, you have choice. How much time it will take? It will take 60 minutes. This also takes 60 minutes, and this takes just 10 to 15 minutes. So nowadays, maximum kits, cDNA synthesis or one-step PCR master mix, they used to use this recombinant MMLV reverse transcriptase. So these are the like major 4-5 differences in all 4 molecules. And the one is what we can think, the yield. The yield with this is medium, with this is low, and with this is high yield. The cDNA synthesis yield is, in this case it is medium, in this case it is low, and in this case is high, because this was modified by keeping 2 points in their mind. One, it can convert longer fragment. Second, we can use higher temperature for cDNA synthesis so that we can prevent cDNA mispriming. What that does means sometimes our primers, they can bind on the non-specific sites and they will start their cDNA synthesis. So to prevent that non-specific cDNA synthesis or priming or false priming, you can say, so in those cases this recombinant MMLV is used to prevent that thing. And the second is, the researcher, they are interested to reduce the time of conversion, because it takes 60 minutes, and because in most of the one-step master mix, we have clubbed cDNA synthesis with your PCR. So in those cases, we want to reduce the time of cDNA synthesis, and also we want to have a high yield cDNA synthesis. High yield we want. So for that, they have incorporated that recombinant MMLV virus, reverse transcriptase from this virus. So this is our choice. We have to think of, we have to select our cDNA synthesis kit, which kit we want to use on the basis of our application. The fourth and most interesting part is, while you are designing your cDNA synthesis, whenever you have selected your reverse transcriptase, okay, the kit is available in your lab, and you want to convert, or you have to, but you have to think of what you have to think of. You have to think of because you want to use. You want to convert your RNA to cDNA. So you have 3 ways. One is you can use your random primers. You can use your random primers to convert your cDNA. These are the random hexamers, different permutation combinations of random hexamers. So the second part is your oligo-DT. Use of oligo-DT primer, and the third one is your gene specific primers. So where to use random primers? Because many times students they confuse with the utility of these 3 type of primers for the synthesis of cDNA. So random primer, where we have to use random primer? The first one is, you have to use where mRNA molecule does not have or poly A tail. जहां पे क्या होगा, पॉली ए टेल, ऐसे mRNA स्पीशीज के साथ हम काम कर रहे हैं जहां पे जिनका cDNA हम बनाना चाहते हैं, बट वहां पे क्या नहीं है, पॉली ए टेल नहीं है. The poly tail is absent in some kind of mRNA molecule, and we are working with those mRNA molecule. In those cases, you have to convert those mRNA molecule with the help of random primers. The second one is, suppose where you have to use random primers. Suppose you are working with rRNA. Your interest is rRNA. You want to see rRNA. So in those cases, you have to convert your RNA molecule, all different type of RNA molecules, to cDNA. So in those cases, you have to use random primer. The third and important thing is, sometime when we have chosen our reference gene 18S, जब भी आप 18S को अपना रेफरेंस और इंटरनल रेफरेंस जीन यूज करना चाहते हैं, that time you have to use your random primers for the cDNA synthesis. क्यों 18S यूज करेंगे, कब यूज करेंगे? एक सिचुएशन है, हम 18S कब यूज करेंगे? जब भी हमारे जीन्स जो हैं, जो हमारे हाउस कीपिंग जीन्स हैं, जैसे बीटा एक्टिन हो गया या माइक्रोग्लोबुलिन हो गया या गैप डीएच हो गया, व्हेन दोज जीन्स आर मॉडिफाइड विद आवर डिजीज कंडीशन और विद द ट्रीटमेंट कंडीशन और वॉट एवर स्टडी यू हैव प्लान, अगर वो सारे जीन्स उसमें मॉडिफाई हो रहे हैं या उनका एक्सप्रेशन अफेक्ट हो रहा है, in those cases you have to select your reference gene as 18S, and for the 18S expression you have to convert your cDNA with the random primer. Then oligo-DT कहाँ यूज करेंगे? जहां पे आपका पॉली ए टेल होगा। कहां पे यूज करेंगे oligo-DT? जहां पे हमारा पॉली ए टेल होगा, 1. And the second is because you want to just convert your mRNA molecules with poly A tail to cDNA. आप दूसरे RNA molecule जो आपके इसके साथ एक्सट्रैक्शन में आए हैं, you don't want to convert those RNA molecule to cDNA. So to increase the specificity. तो जब भी आप जीन एक्सप्रेशन स्टडीज करेंगे और इफ दे आर हैविंग poly A tail at the 3' end, in those cases you have to use oligo-DT primers. तो oligo-DT primer से आप अपना cDNA बनाएंगे। तो आपका ये हो गया। अब थर्ड इज जीन स्पेसिफिक। सो एवरीबडी नोज दैट जीन स्पेसिफिक इज, suppose if I want to study gap DH, so I will use our forward and reverse primers for the cDNA synthesis. Usually the first strand is done by your reverse primer और वो आपका cDNA synthesis में आपका help करता है। So cDNA synthesis जो होगी जीन स्पेसिफिक प्राइमर में, फर्स्ट स्टेप जो होगा वो रिवर्स प्राइमर से होगा, and then it will be carried out by second strand and then complement, then PCR, then amplification cycles with the use of both primers. But आपका जो cDNA होगा वो फर्स्ट स्टेप जो है वो आपके रिवर्स प्राइमर से ही होगा जीन स्पेसिफिक PCR में। जीन स्पेसिफिक जो प्राइमर है या cDNA सिंथेसिस है, यूजुअली हम लोग जब भी हम वन स्टेप PCR परफॉर्म करते हैं उस टाइम पे हम जीन स्पेसिफिक ही cDNA सिंथेसिस करते हैं। तो आप इस तरह से you have to think of whenever you are converting your cDNA, all the possibilities, all the selection criteria. Yes, my RNA is okay, the quality of RNA is maintained and the quality is good. Now then you have to remove the DNA which is the main contaminant in your RNA sample, तो आपको उसको remove करना है। Then you have to select your kit, the reverse transcriptase enzyme, which enzyme you want to use. You want to go with the 15-minute cycle, or you want to go with the 1-hour cycle, or you want to have a high yield sample, or whatever you want to use, you have to select. Then third is which primer you want to use for the conversion of cDNA synthesis. You want to go with the random primer or oligo-DT primer or with the gene specific studies. So all those things you have to think of while you are planning your cDNA synthesis. So एक लास्ट में लोग पूछते हैं या स्टूडेंट्स दे हैव क्वेरी इन देयर माइंड दैट क्या हम cDNA को जेल पे देख सकते हैं? See, the cDNA, if you load cDNA on your gel, you will get smear, because cDNA is not your amplification, it is just conversion, 1-to-1 conversion. So suppose if you have 1000 molecule mRNA molecule, the 1000 molecule, if I will say cDNA synthesis is 100%, then all 1000 molecule will converted into cDNA 1000 molecule only. So it is not amplification, it is conversion. So you cannot see on the gel, you can see if you load on the gel, it will be like smear on the gel. तो आपको किस फॉर्म में दिखेगा? स्मेयर फॉर्म में। You cannot interpret anything from that gel. So don't do that practice. So the good practice is check your RNA quality on the gel if you are working with human sample, but if you are working with the viral samples, then it is very difficult to see your RNA even on the gel. So it's better to convert to cDNA and go with the studies, or even you can go with the nanodrop, or your measurements like 260 by 280 ratio, and your concentration and your amount by nanodrop, or using nanodrop and many other measurement tools. So this is the cDNA synthesis which we have to think of. You can use your kits, but you have to take care all those things while you are planning cDNA synthesis. This is all about cDNA. And one of candidate, they have given me one question that RNA, if RNA is isolated from the plants, so plant, if in the plant also the same thing we have to be take care. In plants, most of the RNA, they are having poly A tail, but the poly A tail is very short, 40 to 50 nucleotide. And in those cases, because whenever the poly A tail is very short, the stability is less. So if poly A tail is longer, then stability is higher. So with the, if you are working with the plant mRNA or plant gene expression studies, in those cases you have to be very careful about your RNA, and you can use any of these primers for the conversion. Thank you.